Hi everyone, in this video I am going to explain you about chromosomes in detail. Chromosomes. At the time of cell division, the chromatin material get condensed to form chromosomes. Thus chromosomes is highly condensed form of the chromatin. Chromosomes are not visible during interphase stage, but during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromosomes can be best studied at metaphase stage because size of chromosome is the shortest during metaphase. Shape of chromosome is studied at Anaphase stage. Generally, at the time of cell division, chromatin material get condensed into chromosomes. Thus, chromosome is highly condensed form of chromatin. Chromosomes are not visible during interphase. Chromosomes can be best studied at metaphase stage. The shape of chromosome is studied at anaphase stage. Chromosome numbers in meiocytes diploid 2n and gametes haploid n of the some organisms. Human beings. Meiocyte 46 chromosomes, gamete stage 23 chromosomes. Housefly meiocyte 12, gamete 6. Rat meiocyte 42, gamete 21. Dog meiocyte 78, gamete 39. Cat meiocyte 38, gamete 19. Fruit fly meiocyte 8, gamete 4. Ophioglossum, a fern which contains highest number of chromosomes in living world, 2n. Our diploid condition is 1260, in gamete 630. Apple meiocyte 34, gamete 17. Rice meiocyte 24, gamete 12. Maize meiocyte 20, gamete 10. Potato meiocyte 48, gamete 24. Butterfly meiocyte 380, gamete 190. Onion meiocyte 16, gamete 8. The number of chromosomes in a gamete is called genome, or a complete set of chromosomes inherited as a unit from one parent is known as genome. A single human cell has approximately 2 meter long thread of DNA distributed among its 23 pairs of chromosomes. The number of chromosomes in a gamete is called genome, or a complete set of chromosomes in an inherited unit also called genome. Human beings, each cell contains 2 meters or 2.2 meters of DNA, which is distributed in 46 or 23 pairs of chromosomes. Types of chromosomes. On the basis of position of centromere, chromosomes are broadly classified into 4 types: Telocentric chromosomes, Acrocentric chromosomes, Metacentric chromosomes, and submetacentric chromosomes. Telocentric chromosomes. When centromere is terminal or located at the tip of chromosome called telocentric chromosome. Acrocentric chromosome, when the centromere is subterminal or located near the tip, is called acrocentric chromosome. Metacentric chromosome, when the centromere is located at the mid of the chromosome. Submetacentric, in this type, centromere located near the center or midpoint of chromosome. Telocentric chromosome, centromere is terminal. In acrocentric chromosome, centromere is subterminal or located near the tip. Metacentric chromosome, centromere is located at exactly middle or center. In submetacentric chromosome, centromere located nearly center or midpoint of chromosome. Let's see the diagrammatic representation of types of chromosomes based on the position of centromere. Very first one, metacentric chromosome, in that centromere is exactly middle. In submetacentric chromosome, centromere is just away from the middle or mid of the chromosome. In acrocentric chromosome, centromere is at one side of chromosome. In telocentric chromosome, Centromere is completely terminal. Let's check these diagrams carefully for your better understanding of types of chromosomes based on the position of centromeres. Karyotype. Karyotype is external morphology of all chromosomes of a cell which is specific for each species of living organisms. Karyotype can be studied in metaphase of mitosis. Karyotype includes the number of chromosomes, relative size, position of centromere, length of the arms, secondary constrictions, and banding patterns. Idiogram. The diagrammatic representation of karyotype is called idiogram. In idiogram, chromosomes are arranged in decreasing order of size. Sex chromosomes are placed in last. Idiogram in specific for every species. Karyotype, the external morphology of all chromosomes of a cell which is specific for each species is called karyotype. Diagrammatic representation of karyotype is called idiogram. Structure of chromosomes. Chromosomes shows following structures. Very first one, chromatid. At metaphase stage, each chromosome is consists of two cylindrical structures called chromatids. Both sister chromatids are joined together by a common centromere. A chromosome may have single chromatid in anaphase or telophase, two chromatids in prophase and metaphase. Each chromatid is consists of single long thread of DNA which is associated with histone. Non-histone proteins are RNA and RNA are also present in chromatid. Chromatid, generally we can see that in metaphase stage, chromosome with single chromatid in anaphase or telophase, chromosome with two chromatids in metaphase. Each chromatid contains one long thread of DNA with histone, and non-histones and RNA also present in chromatid. Number 2, centromere. Each chromosome at prophase or metaphase is consist of two chromatids. Both the chromatids of a chromosome are joined or connected by a structure called centromere. At center, two protein discs are present which is called kinetochore. Kinetochores constitute the actual site of attachment of spindles to chromosomes during cell division. At the region of centromere, the chromosome is comparatively narrower than remaining part of chromosome. Thus, it is termed as a primary constriction. Each chromosome is consist of two chromatids. Both chromatids are joined or connected by a structure called centromere or primary constriction. The centromere covered by proteinaceous disc-shaped structures called kinetochore. Kinetochore is the exact structure where the spindle fibers are connected to chromosomes. At the region of centromere, the chromosome is comparatively narrower than remaining part of chromosomes. Thus, it is termed as primary constriction. Let's see the diagrammatic representation of chromosome with the kinetochore. At center, centromere is present which connects two chromatid bodies. At the same time, these centromere covered by proteinaceous disc-shaped structures called kinetochores. These kinetochores are the exact places where the spindle fibers are attached at cell division stage. Secondary constriction. Besides primary constrictions, other constrictions may also occur on some chromosomes which are known as secondary constriction. These constrictions are non-staining and found at constant location. Secondary constriction is also known as NOR, nucleolar organizer region. 13, 14, 15, 21, 22 chromosomes in human beings are present at secondary constriction region. Secondary constriction. Besides primary constrictions, other constrictions may also occur at some places of chromosomes which are called secondary constrictions. These secondary constrictions are also called NOR, nucleolar organizer region. Telomere. Chromosomes have polarity and polar ends of chromosomes are known as telomeres. Telomeres prevent fusion of one chromosomes to other chromosomes. Telomere rich in guanine bases. Enzyme telomerase synthesizes telomere part of chromosome, which is ribonucleoprotein. Telomeres of chromosomes becomes shorter during aging process. Telomeres. Chromosomes have polarity and polar ends of chromosomes, which are called telomeres. Telomeres prevents the fusion of one chromosome with another. Telomeric region is rich in guanine bases. Enzyme telomerase synthesizes telomere part of chromosome, which is a ribonucleoprotein. Telomeres of chromosomes becomes shorter during aging process of chromosome. Special type of chromosomes. Salivary gland chromosomes. This type of chromosomes was discovered by E.G. Balbiani in larva. This chromosome is called polytene chromosome because number of chromatids are very high. Swollen areas present at some places in polytene chromosome, which are called as Balbiani rings or puffs. These puffs helps in synthesis of RNA and proteins. Salivary gland chromosomes concerned with metamorphosis and molting process of insect larva. Salivary gland chromosomes are generally present in saliva of insect larvae, first discovered by Balbiani. So these are also called Balbiani rings or polytene chromosomes. Salivary gland chromosomes concerned with metamorphosis and molting process of insect larva. Second one, lamb brush chromosomes discovered by the scientist Flemming and Ruckert from oocytes of vertebrates, amphibia. During diplotin stage of cell division, these chromosomes are look like lamb brush, thus called as lamb brush chromosomes. Axis of lamb brush chromosome is consist of DNA, while matrix is consist of RNA and proteins. Lamb brush chromosomes is concerned with vitellogenesis or yolk formation. Lamb brush chromosomes discovered by Flemming and Ruckert, very first in oocytes of vertebrates, particularly during diplotin stage of cell division. Lamb brush chromosomes is concerned with yolk formation. Axis of lamb brush chromosome is consists of DNA, while matrix is consists of RNA and proteins. These lamb brush chromosomes are look like lamb brush or test tube brush. Up to here, the chapter Cell, the Unit of Life was completed. In next video, I will start new chapter. Thank you very much.